Hi guys, Mrs. Horn here. Today you're going to spend time working on an activity with enzymes, different from last week's lab because our focus is on the shape of the enzyme and why is it so important. What we haven't talked about in depth yet is that the shape of an enzyme, we know it's specific, but how does it get that shape? And it gets that shape because the DNA, the code for DNA, told it to be that shape. DNA made it the shape that it is. So there's three main parts to the activity that you're going to do. The first one is you're going to make a guess. You're going to be given a substrate, and you have to make a guess as to what that enzyme could look like that would fit it. Then the second part is I'm going to give you directions as to how to build the real enzyme, and then you're going to build that, and it should, if done the right way, fit in with the substrate. And then the third part is what happens if that DNA was mutated? What if there was a little mistake in the DNA? Would that lead to different components that make up that amino acid or that make up the enzyme? And if you're using different amino acids for the enzyme, would it change its shape? And if it does change its shape, is it still going to work? In this activity, in the lab today, or in the activity, you're going to be using Legos. Legos will be your substrate. Legos will also be your enzyme. Once you have your blue tray, on the tray will be a bunch of materials. You'll be using everything on the tray. You will have colored sheets of cardstock that's, let's see, you should have one that looks like this. It's a packing list. You should have one that explains the shapes of the Legos. If you are not a master builder, this will be helpful to you. And you will also have a sheet called the blueprint. This will come in very handy when you are building your enzyme. This is also a good sheet to refer to up on the screen here because it's in color. The copy that you have on your tray is not so easy to understand because the color didn't come out. So refer to this when you need to kind of compare your build structure to what it actually should be, because this is the only way I can figure out what it's supposed to look like. You also have a bag of loose Lego pieces. The bag is what you will use to build your hypothetical enzyme, your normal enzyme, and 4 mutated enzymes. There's also a Dixie cup on your tray with a structure already built. That's the substrate. You should not take the substrate apart, leave it as is. You will also need a yellow packet that looks like this. On the top of this yellow packet, I need you to take some notes. As you get further through the activity, you will be transcribing and translating DNA into RNA into amino acids. This information I have in blue right here will come in very handy. This is a skill you probably have learned in middle school, you probably learned it in living environment, but it's always nice for a refresher. In DNA, nitrogenous bases have a complementary pair. They have a complementary base. So G always goes with C, and A always goes with T. Easy way to remember this, Greece Central, Arcadia Titans. When you're working with RNA, the only difference is that RNA does not have a T, it has a U for uracil. My recommendation for you to get through this activity, because you're going to be very student-driven, is to read the introductory information, read this background. The part 1 procedure is laid out for you, tells you exactly what to do. Be sure to do things in the order that it is explained. Do not jump to conclusions and do not skip ahead to something that you think you need to do, because you might miss making a prediction or miss making a sketch. So please follow along in the order as it is given. The second part of the lab is where you will be creating your mutant enzymes. There are 4 of them. There's also a worksheet page that you will be using for both part 1 and part 2. And there is, can't find it. That's not it. This genetic code chart, which is in your yellow packet, but there's also I think a purple copy of it on your folder or on your tray. This chart here will come in very handy when you're filling out your worksheet. This is the worksheet page. There are some post-lab questions that need to be completed. I would like you to write those on a separate sheet of paper. I am collecting this at the beginning of the period. So part 1 must be completed, part 2 must be completed, the worksheet page has to be done, and the post-lab, the post-activity questions should be done in complete sentences on a separate sheet of paper. I'm going to leave the last screen on this page so that you have a colored picture of this blueprint page. You have a copy of it, but you don't have it in color. So if you need to look back at this, feel free to pause the screen here so that you can see what it looks like. Colors that you have of your Legos are not going to match this, and that doesn't matter. Color doesn't matter. We're only going for shape here. So good luck on the activity, and I hope this helps.